In this unit, I'll talk about bacteriophages and biochemistry. There's a lot of material in this unit, and these are some important learning objectives that you should keep in mind as you go through it. You should identify the 4 important biological polymers and be able to recognize their monomeric parts. Understand the major roles for each of these 4 polymers in biology. Describe the 4 levels of protein structure interaction. Understand the chemical properties of amino acid side chains. Draw the basic structure of an amino acid and nucleotide using R for the side chain or nitrogen base. Draw the hydrogen bonding interactions between 2 nucleotides when given the chemical structure of the nitrogen bases. Write complementary DNA sequences correctly, identifying the 5' and 3' ends of the complementary molecules. Describe the makeup of bacteria cell membranes and cell walls and how the chemistry of molecules involved lead to their function. Describe how different phages penetrate the cell wall and cell membrane of bacteria. And then describe 3 ways we learn about the structure of biological molecules. Finally, we'll talk about one important experiment in biology that was done using phages. This is the Hershey and Chase experiment. You should understand this experiment and describe how isotopes allow detection of proteins and nucleic acids. Describe how blending and centrifugation affect the phage and bacteria. Interpret data and draw conclusions from this experiment. Then you should finally be able to explain how the Hershey and Chase experiment identified DNA as the genetic material. So in part 1, we'll talk about polymers, and the first 2 polymers that we'll cover specifically are carbohydrates and lipids. So these 4 molecules, or types of molecules, carbohydrates, lipids, proteins, and nucleic acids, are the important biological macromolecules. So macromolecules are molecules of a substantial size, and these macromolecules are what we call polymers. Polymers are chains of repeating molecular units. Those repeating units are called monomers. So in any molecule, any molecule consists of atoms that are connected together by covalent bonds. So the covalent bonds in polymers, there are covalent bonds that connect together the atoms that make up the monomers, and there are also covalent bonds that connect the monomers to make the polymer. So one of the things you want to pay attention to in this unit is that the monomers have certain chemical patterns that you should be able to recognize. And then the chemical bonds, the covalent bonds that join together the monomers are also important. You should be able to recognize those. So all 4 biological polymers that we will discuss are made via a condensation reaction. Remember from the last unit that this is a reaction where water is removed from the reactants to create, in the process of creating a new covalent bond. So you can think about the structure of monomers and polymers using these pictures as a pattern. Right? So the monomeric units are the circles in the branch chain polymer down here on the bottom. You can see there are bonds between those repeating circle units. Those bonds are sometimes regular patterns, and sometimes there are branch points or other interesting connections between these monomers. The other important thing to understand about monomers is that they are made of many atoms, and so this simple diagram gives you an idea that a monomer is a small molecule, so it's got some repeating atomic parts, and then in the polymer, those monomeric units are joined together. So the first polymers that we'll talk about are carbohydrates. These are molecules that have many important functions in the cell, including serving as important structures and also as important sources of energy. Carbohydrates consist of carbon, hydrogen, and oxygen with a repeating formula CH2O. So carbohydrates come in 3 different flavors. There are monosaccharides, disaccharides, and polysaccharides, and like the name suggests, monosaccharides consist of 1 sugar ring. Disaccharides consist of 2 monosaccharides joined together, and polysaccharides are chains of many monosaccharides or disaccharides that make up these long polymer units. So one of the things you should do after last unit is recognize some of the common chemical elements of these molecules. So we said that these molecules consist of repeating units of carbon, 2 hydrogens, and oxygen. So let's go through this molecule of glucose here and count the carbons, hydrogens, and oxygens. Here we have the carbons numbered for us. So there's 1, 2, 3, 4, 5, 6 carbons in this molecule. There are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens. So there's twice as many hydrogens as there are carbons. And then you can see all the oxygens. There's 1, 2, 3, 4, 5, 6 oxygens. Okay. So you have this repeating formula. There's twice as many hydrogens as oxygens and an equal number of carbons. Okay. So disaccharides are when 2 monosaccharides are joined together. And when any monosaccharide or disaccharide is joined together to make polysaccharides, we have a condensation reaction. Right? So one of the hydroxyl units on this molecule is water is removed from those molecules, and that forms a covalent bond between those 2 monosaccharides. Right? So this is one of those important roles for water. It plays a role in many chemical reactions inside the cell. The other thing that you should notice after having gone through Unit 3 are some of the common chemical properties of these molecules. Right? So think about things like how hydrophobic or hydrophilic some of these molecules might be. We can see on this structure of glucose over here several important functional groups. In fact, there are hydroxyl units at every single carbon position on this ring structure. Right? Those hydroxyl units are polar functional groups, and that makes carbohydrates generally pretty water-soluble or hydrophilic. So monosaccharides and disaccharides. Monosaccharides are formed, they are rings of repeating units of carbon, hydrogen, and oxygen. And disaccharides are formed when 2 monosaccharides are joined in a dehydration synthesis, or what we've been calling a condensation reaction. Those are 2 interchangeable terms. So disaccharides can be broken down by hydrolysis. Right? Again, important role of water in biology is participating in these chemical reactions. Right? The units of monosaccharides are joined together to form disaccharides and polysaccharides by removing water and forming a covalent bond between those monomeric units. And then in breaking down those disaccharides and polysaccharides, that chemical reaction is reversible and water can be chemically added back to that bond to break the covalent linkage between the 2 monomer units. So when we, in our bodies, build up carbohydrates like glycogen to store energy inside of our cells, right? That is releasing water in the process of building those complex carbohydrates. And when we digest carbohydrates, such as starch when we eat food, that requires a lot of water because water is added as a reactant to break down those chains of monosaccharides. So polysaccharides they consist of chains of tens or hundreds of either monosaccharide repeating units or disaccharide repeating units. So monosaccharides and disaccharides can be the monomer units for polysaccharides. So starch, glycogen, dextran, and cellulose are all polymers of glucose. And the different ways the monomers in these carbohydrate polymers are linked together can change the chemical structure, right? And that changes the chemical properties. So these are all polymers of glucose, but they all differ in their bonding. and that also causes them to have different functions in the cell. So amylose, or starch, is a long linear chain of glucose that's bound together in a structure like this shown on the left. And you can kind of draw a cartoon of this using this repeating hexamer shape right here. Right? So amylose is a long linear chain. It's reasonably water soluble, but it's a relatively simple chain of glucose. Starches are more complex carbohydrates. Right? They include many more bonds. They include these chains of glucose, like we have in amylose. And they also have branch chains where there are additional bonds on some of these additional hydroxyls that are part of the ring of the glucose monomeric unit. Right? So these branch chains make this polymer of glucose. It makes it a bit more complicated. These molecules can get tangled in each other. It makes them more sticky because they get those chemical tangles. And because they're more sticky, more complicated molecules, they're a little bit less water soluble. Glycogen is a more complex branch form of these carbohydrates, right? So we don't have just one central long chain of glucose joined together and glucose branches coming out from that. We also have branches on the branches, right? So we have a lot of covalent permanent bonds between these monomeric linkages, right? To break down this chemical, like glycogen, you have to use lots of water to break down these individual covalent bonds between the monomeric units, right? And so glycogen takes longer to break down. It's less water soluble. And glycogen is used by our cells and in other cells in order to store energy because it includes many monosaccharide units that are packed tightly together. Cellulose is the most bonded and the most complex molecule. Right? We don't have just chains of glucose joined together by covalent bonds that look like this. Each one of those glucose monomers also has an additional covalent bond to form this row and lattice. Right? So cellulose is more commonly referred to as fiber. It is the carbohydrate that makes up the walls in plant cells, the cell walls. It is a pretty strong, stable, sticky molecule because of all of these additional covalent bonds that are joining those monomeric units together. Right? So the pattern of bonding between different types of carbohydrates changes their chemical properties. So the next of the four biological macromolecules that we'll talk about are lipids. So lipids, the primary important role for lipids is they are the primary components of cell membranes. They also have a number of additional important cellular functions, including they function as an energy store, so we use fats as energy. They also form signaling molecules like steroids. And there are a number of additional places where you'll see fats in biology. The important role of lipids comes from their chemical property that they are generally very nonpolar. So they have no charge and they are insoluble in water, so they're hydrophobic. So when we think about lipids, a simple lipid is what we call a fat or a triglyceride. Right? And so these fats consist of monomeric units where we have two distinct monomers. We have a glycerol monomer and we have a fatty acid monomer. The glycerol monomer looks a little bit like a carbohydrate, but it's not. But it does have these hydroxyl groups on the end of it. The fatty acid consists of a hydrocarbon chain. So this is a chain of carbons covalently bonded together in a linear form, with hydrogen decorating those carbons. Remember that these carbons evenly distribute charge, unlike the electrophilic oxygen. And so these hydrocarbon chains are extremely nonpolar and extremely hydrophobic. The glycerol and the carboxyl or carboxylic acid N, this is the one functional group on the fatty acid. These are more polar groups, and these are a little bit more water soluble than the rest of the hydrocarbon chain. Just like in carbohydrates. Right? So we have these hydroxyl groups on the end of glycerol and an OH on the end of the carboxylic acid group in the fatty acids. And these can join together, these monomeric units, by this dehydration synthesis or condensation chemical reaction. Now, triglycerides are when we have 3 fatty acids that are all covalently linked to a single glycerol. So one thing that you may have noticed about glycerol is that there are exactly 3 hydroxyl groups on glycerol. And that means that there are 3 opportunities for things to be linked to glycerol by these condensation reactions. When those 3 things are all fatty acid chains, right, that is a triglyceride. Now, triglycerides and fatty acids, one important thing to notice about them. So we have fatty acid chains that have different lengths or different number of carbons. We also have fatty acid chains that include double covalent bonds. Now, the double covalent bond plays an important role in the structure and chemical properties of fats and things that contain fatty acids. Right? So the single covalent bonds, they are relatively flexible because it allows those atoms that are joined together to rotate together in a number of different ways. The double covalent bonds, they are more rigid, so they have there's less freedom for those covalently bonded carbons that are joined by the double bond to move around, and so this creates a kink in the structure of these fatty acids that contain a double covalent bond. Right. So we call these saturated fatty acids, right? And unsaturated fatty acids. Saturated fatty acids are saturated with hydrogen. So if those fatty acids all contain single covalent bonds between the carbons, right, that means that carbon forms 4 covalent bonds, and since there are 2 with other carbons in the chain, there are also 2 hydrogens bonded to each one of those carbons in the chain. So it's got the maximum number of hydrogens on it. It's saturated with hydrogens, is a saturated fatty acid. The unsaturated fatty acids are the ones that contain these double covalent bonds. Right? So when we have the double covalent bond, there's 2 covalent bonds joining these carbons together. That means rather than having 2 hydrogens, they each can have 1. And so rather than having the maximum number of hydrogens in this fatty acid chain, we are missing 2. And so it's unsaturated with hydrogen. Okay? Unsaturated fatty acids are kinked, have double covalent bonds, and fewer hydrogens. Saturated fatty acids are all single covalent bonds and have the maximum number of hydrogens. So what role does this saturated and unsaturated fatty acid play in the biology of these different fats? Right. So if there are a large number of saturated fatty acids in the molecules in a substance, right? Those saturated fatty acids, those chains are flexible. They can pack together tightly in substances. And that means the melting point of substances that contain lots of fatty acids, contain lots of saturated fatty acids, things like butter, they are more solid at room temperature. It takes a higher temperature to melt them. Foods and substances that contain a large number of unsaturated fatty acids, right, that have those double bonds those kinks in the chain, those don't pack as well together because of those double bonds. Right? And that means that things that contain lots of them are more liquidy. They melt at a lower temperature. Right? So things like olive oil that contains lots of unsaturated fatty acids melt at a lower temperature. This is important to keep in mind when we get into the structure of membranes, because membranes have a balance of saturated and unsaturated fatty acids to keep the membrane the right amount of liquid for things to move around in it. So complex lipids are lipids that contain 3 different monomeric parts. Right? So we have the glycerol and the fatty acids, just like in the triglycerides. And we also are going to throw on a phosphate group. Phospholipids are important because they have both highly polar regions and very non-polar regions. Right, so this is the structure of a phospholipid. So we have the glycerol, it's got those hydroxyls on it. This forms the center part of this polymer. Our fatty acids, either saturated or unsaturated, they are joined to the glycerol at those hydroxyls through a condensation or dehydration synthesis reaction. And then, rather than a third fatty acid chain, here we have a phosphate group. Right? The phosphate group can have a number of things additionally attached onto it. But the phosphate group in general, it contains one of these 6 important biological atoms, phosphorus. Right? It has lots of oxygens on it, and phosphates tend to be negatively charged. Right? That means that in a regular solution of water, they have this negative charge, and that makes them very polar and very hydrophilic. So we have these non-polar hydrophobic hydrocarbon chains and these hydrophilic water-loving phosphate group ends. This allows these phospholipids to form the phospholipid bilayer that makes up membranes. Right? So the inside and outside of the cell, the area that cells work in, is generally composed of a lot of water. Right? So we have water on the inside of the cell, water on the outside of the cell. The hydrophilic phosphate end of this molecule interacts very well with water. The hydrophobic end of the molecule, it doesn't interact with water, and it interacts with other nonpolar substances. So the phospholipid bilayer is made by these layers of 2 phospholipids where the nonpolar chains are interacting on the inside of the membrane lipid bilayer, and the phosphate ends, the hydrophilic ends, are on the outside. Now, if membranes have more saturated fatty acids, they pack closer together, and that makes these membranes more solid. And if they have more unsaturated fatty acids, those are less well packed and more liquidy. So cells need to balance the amount of saturated and unsaturated fatty acids in order to keep their membranes balanced just right. There are many other important examples of complex lipids in biology. So lipopolysaccharide, which is also known as LPS, is an important molecule found in the membranes of gram-negative bacteria cells. Here we have 2 glycerols with fatty acid chains covalently bonded to them. And then rather than being bonded to a phosphate, there's actually a polysaccharide, right? All of these carbohydrates that are attached to the end of one of those glycerols, they form an important trigger for our human immune system in recognizing gram-negative bacteria. We also have lipoteichoic acids. Those are also carbohydrates that are bonded to a lipid that help anchor the cell membrane of gram-positive cells to the cell wall. There are other types of lipids as well, including steroids. So steroids, the ring structure of steroids is quite a bit different from lipids. They do consist of these hydrocarbon chains, but instead of being the long chains that you see in fatty acids, they form these ring structures. So steroids are super hydrophobic. They don't mesh well in water. They do mix well in cellular membranes. And so steroids actually form what we call lipid rafts, which float in the membranes of many organisms. Steroids also have an important function as hormones or signaling molecules in mammalian organisms, including estrogen and testosterone. And so these are molecules that induce many important biological processes, including many of the biological processes that go on in developing sex differences between individuals during puberty and during development.